All right, everybody, here is video 2 going through the slides from meiosis. So in video 1, we just kind of went over the generalities of what is meiosis and why do we care. So now we're going to start going through meiosis 1, because remember there are 2 rounds in meiosis. Okay, so just like mitosis, we have to start with chromosome replication. You have to double up that DNA or there won't be enough to go around in the end. So DNA replication has to come first. Now we go into prophase 1. So in prophase 1, the DNA coils into chromosomes, same as we've seen before. You got to get that DNA coiled up so it stays nice and organized, you're not losing bits. And you have to get it to your destination, and so coiling it up keeps everything nice and organized. Then the spindle fibers will appear, so that's what's going on here. And these lines coming down, these are microtubules, which are proteins created by the cells, and that keeps everything organized. Now the homologous chromosomes line up together. So now homologous chromosomes, you got one from your mom and you got one from your dad. They have DNA for the same genes. And so these chromosomes that are lined up here, purple is from one parent, orange is from the other. They line up together and they have DNA for the same genes. So these might both have the DNA for eye color and they are together. So each pair of chromosomes is also called a tetrad, and this pairing is called synapsis. So now we have crossing over. So what you're going to see is some of the purple DNA is going on to the orange chromosome and vice versa. So this is really quite an amazing process if you think about it, because it doesn't make mistakes. It lines up and it swaps this DNA very nicely in an organized fashion. And this is our first source of genetic variation. So this is really important, is this first source of genetic variation. Okay, so if you look here in this picture, it'll probably be a lot easier if you look at it on the slides itself rather than in this video, but you're going to see that there are letters on these chromosomes. And what it's trying to show you, imagine each letter is a different gene. Look how nicely they swap. We're not getting bits of I in with the J. It's swapping very nicely. So this is also called genetic recombination. Another way of saying we are swapping up this DNA, getting that variation which species need in order to adapt and continue on. Don't want to end up like those cheetahs in the article I gave you. Now we have metaphase 1. So remember, metaphase and mitosis, metaphase meet in the middle. So there's that metaphase plate going across the middle of the cell, and that's where the chromosomes line up. But what's different in meiosis in metaphase 1 is that the pairs are lining up together side by side. And at this point, the spindle fibers will attach to the centromere, which is the middle of the chromosome. So it's represented by that circle there. That's then the spindle fibers just keeping things organized. So now we have anaphase 1. So anaphase, remember anaphase, away. These chromosomes are getting pulled apart. But we're pulling whole chromosomes to the opposite poles of the cell. We're not breaking up those chromosomes yet. It's whole chromosomes. So notice that the purple and the orange, the purple doesn't have to stay with purple and orange doesn't have to stay with orange. Remembering those are from different parents. They can be sorted in any order. So it's random separation of those chromosomes. It's called independent assortment. This is the second place where you get significant genetic variation through this process. So this is also really important. Gotta have everybody being different. So now we have telophase 1 and cytokinesis 1. So in telophase, these chromosomes are getting to the opposite poles. each of these chromosomes, though, notice they're still in that X form. So they have 2 chromatids still. And then cytokinesis will occur. So the one cell is becoming 2, but we still have doubled up DNA there. And next video, we're going to start here. Email me with questions.